The role of cell adhesion receptors in transmembrane signaling. One of the roles of integral membrane proteins is to transfer information across the plasma membrane, a process known as transmembrane signaling. Transmembrane signaling. While the subject will be explored in detail and later in the next chapter, it can be noted that all four types of cell adhesion molecules have the potential to carry out this function. Culture dish, along with that there will be focal adhesion, and in between this the hemidesmosome can be seen, and Catherine interaction, IGSF and IGSF interaction, integrin IGSF and selectin lectin interaction. An overview of the types of interaction involving the cell surface. 4 types of cell-cell adhesion interactions are shown, as well as 2 types of interaction between cells and extracellular substrata. Keep in mind that the various interactions depicted here do not occur in connection with a single cell type, but are shown in this manner for purpose of illustration only. For example, interactions between selectins and lectins occur primarily between the circulating leukocytes and walls of the blood vessels. Catherines can transmit signals from the extracellular environment to the cytoplasm by means of linkages with the cytoskeleton and with cytosolic regulatory molecules such as protein kinase and G proteins. Protein kinase activate or inhibit their target proteins through phosphorylation, whereas G protein activate or inhibit their protein targets through physical interactions. The engagement of an integrin with its ligand can induce a variety of responses within a cell, including changes in cytoplasmic pH or Ca2 plus concentration, protein phosphorylation, and gene expression. These changes, in turn, can alter a cell's growth potential, migratory activity, state of differentiation, or survival. This type of phenomenon is illustrated by the mammary gland epithelial cells shown. When these cells are removed from a mammary gland and grown on a bare culture dish, they lose their ability to synthesize milk proteins and appear as flattened, undifferentiated cells. When these same undifferentiated cells are cultured in the presence of certain extracellular molecules like laminins, they regain their differentiated appearance and become organized into milk-producing gland-like structures. Laminin is thought to stimulate mammary cells by binding to cell surface integrins and activating kinase at the inner surface of membrane. So, the role of extracellular protein in maintaining the differentiated state of cells. The mammary gland epithelial cells were removed from a mouse and cultured in the absence of extracellular matrix. Unlike normal differentiated mammary cells, the cells are flattened and are not engaged in the synthesis of milk proteins. When extracellular matrix molecules are added back to the culture, the cells regain their differentiated appearance and synthesize milk proteins.